Excretory products and their elimination. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium ion, potassium ion, chlorine, phosphate, sulfate, etc., either by metabolic activities or by other means like excess ingestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanisms of elimination of these substances with special emphasis on common nitrogenous wastes. Ammonia, urea, and uric acid are the major forms of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with the minimum loss of water. The process of excreting ammonia is ammonitolism. Many bony fishes, aquatic amphibians, and aquatic insects are ammonitolic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surfaces or through gill surfaces in fish as ammonium ions. Kidneys do not play any significant role in its removal. Terrestrial adaptation necessitated the production of lesser toxic nitrogenous waste like urea and uric acid for conservation of water. Mammals, many terrestrial amphibians, and marine fishes mainly excrete urea and are called urotelic animals. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, land snails, and insects excrete nitrogenous waste as uric acid in the form of pellet or paste with a minimum loss of water and are called uricotelic animals. A survey of animal kingdom presents a variety of excretory structures. In most of the invertebrates, these structures are simple tubular forms, whereas vertebrates have complex tubular organs called kidneys. Some of these structures are mentioned here. Protonephridia or flame cells are the excretory structures in platyhelminthes, flatworms, example planaria, rotifers, some annelids, and the cephalochordate Amphioxus. Protonephridia are primarily concerned with ionic and fluid volume regulation, that is, osmoregulation. Nephridia are the tubular excretory structures of earthworms and other annelids. Nephridia help to remove nitrogenous waste and maintain a fluid and ionic balance. Malpighian tubules are the excretory structures of most of the insects, including cockroaches. Malpighian tubules help in the removal of nitrogenous waste and osmoregulation. Antennal glands or green glands perform the excretory function in crustaceans like prawns. Human excretory system. In humans, the excretory system consists of a pair of kidneys, one pair of ureters, a urinary bladder, and a urethra. Kidneys are reddish-brown, bean-shaped structures situated between the levels of last thoracic and third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in width, 2 to 3 cm in thickness, with an average weight of 120 to 170 gram. Towards the center of the inner concave surface of the kidney is a notch called hilum, through which ureter, blood vessels, and nerves enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis with projections called calyces. The outer layer of kidney is a tough capsule. Inside the kidney, there are two zones, an outer cortex and an inner medulla. The medulla is divided into a few conical mosses, medullary pyramids, projecting into the calyces. The cortex extends in between medullary pyramids as renal columns called columns of Brittany. Each kidney has nearly 1 million complex tubular structures called nephrons, which are the functional units. Each nephron has two parts, the glomerulus and the renal tubule. Glomerulus is a tuft of capillaries formed by the afferent arteriole, a fine branch of renal artery. Blood from the glomerulus is carried away by an efferent arteriole. The renal tubule begins with a double-walled cup-like structure called Bowman's capsule, which encloses the glomerulus. Glomerulus, along with Bowman's capsule, is called the Malpighian body or renal corpuscle. The tubule continues further to form a highly coiled network, proximal convoluted tubule, PCT. A hairpin-shaped Henel's loop is the next part of the tubule, which has a descending and ascending limb. The ascending limb continues as another highly coiled tubule region called distal convoluted tubule DCT. The DCTs of many nephrons open into a straight tube called collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the calyces. The Malpighian corpuscle, PCT and DCT of the nephron are situated in the cortical region of the kidney, whereas the loop of Henel's dips into the medulla. Majority of nephrons, the loop of Henel is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. In some nephrons, the loop of Henel is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephrons. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule called the peritubular capillaries. A minute vessel of this network runs parallel to the Henel's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephrons. Thank you and all the best.